So we've taken a strategic approach to how we prioritize the testing of these samples. And our approach is to prioritize the specimens that have to do with the route of transmission at that interface. So for instance, if the sample that we're testing happens to be a bushmeat sample, the route of transmission would likely be through the blood or the bodily fluid of that animal. So we would test the whole blood, serum, or plasma, or tissue. If the animal was collected around a house, a paradomestic rodent, for instance, the likely source of infection would be through a bite or defecation or urination in the food or around the house. So we'd test oral swabs, rectal swabs, or urine or feces. Yep.
How do you test for an unknown virus if it's unknown? How do you know to test for it?
You set me up for the next slide. So the next step is how to test for an unknown virus if you don't know what it is. So the way we do this is instead of testing for specific viruses, we've developed molecular assays known as polymerase chain reaction or PCR reactions that are designed in such a way that they don't just detect one virus, like West Nile virus, for instance, but instead they detect the entire family of viruses that that virus falls under. So for West Nile virus, that would be flaviviruses. For Ebola virus, it would be filoviruses. And what this allows you to do is, one, you're more efficient in your testing because you don't need to test for every single member of that family. But two, by developing it in this manner, you're able to not just detect known viruses, but other viruses within that family that are unknown. So near neighbors of those known viruses. Additionally, a small subset of our samples are sent out for something called deep sequencing. I may go into that in a little bit more detail later, but essentially it's a form of sequencing of the nucleic acids in a sample that's completely unbiased. So you can take a sample and identify every bit of nucleic acid, so DNA and RNA, within the sample. I'd love to do that with all of our samples, but it's really expensive and takes quite a bit of time. And also, it can't be done in these foreign locations, at least not most of them. It requires a very high-tech laboratory, and so samples generally need to be brought into the US for that. The reason that we don't want to bring the samples to the US is one of the main purposes of this project, as I mentioned earlier, isn't for science, isn't to detect a bunch of new viruses. It's to build up the capacity in these locations so that they can detect and identify these new viruses. That's one of the reasons why we took one of the simpler approaches like PCR, because it's something that you can set up very easily in a large number of locations and push all of that technology out to the field sites so that they can do their testing there, and it leads to a sustainable capacity building in those locations. And this next slide just gives you an idea of the variety of viruses that we test for. Many of these may be familiar, some of them not so familiar, but these include just about every virus family that's been implicated in major human outbreaks throughout time. Does it get quieter when I talk this direction versus this direction, or is it just my hearing of the microphone? I can move this microphone. Okay, hearing nothing, I'll just go on. So in pushing all of this technology out to the field, one of the bigger challenges that we ran into that we didn't anticipate is that for these types of assays, like with almost any test, you need to have a positive control. So when you run the assay, you run it with a negative control and a positive control. And the positive control allows you to see if the assay is working. If you don't do that, then you could run through a lot of samples and think, oh, everything's negative. But it turns out it just wasn't working. You forgot some ingredient that was important. And generally, the way that you would do it with this type of approach, with PCR, is you would send out an aliquot of the virus that you're testing for. You could then process that and use it as your positive control. But for biosecurity and also just logistics, sending out a large number of tubes of highly pathogenic virus across the world wouldn't be the best approach. But also it requires cold chain, and it's quite difficult, as you might expect, to get keep things cold as you move them to these locations. So we've developed a plasmid-based positive control. And a plasmid is essentially a circular piece of DNA. And within that DNA, we've engineered little bits of all of these viruses. This isn't a living virus, so no one can become infected with it. It's completely artificial and has just very small bits of each of these viruses. So what this allows us to do is use a single piece of DNA as a positive control for all of our assays. And the other nice thing about plasmids is they're very stable. If you needed to, you could actually take a drop of it and put it on a piece of paper and send it in an envelope out to the labs. Although we generally do put it in tubes just so it's a little bit more stabilized. Additionally, we've artificially put false codes into it so that if people get a positive, they can then test their positive and ensure that the positive isn't from the positive control. Contamination from the positive control, especially when you're dealing with these sort of stable plasmids, is a big problem. So I believe we actually coded within the nucleic acid, the amino acids spell out predict, the name of the project. So no virus in the world spells that out with their amino acids. It makes it easy to identify those. So once we have all these results, we don't want them to just sit on some computer somewhere and go unused. We want them to benefit public health. And so in addition to partnering with the Ministries of Health or Ministries of Agriculture and Forestry in all the countries where we work, we've also partnered with a group out of Boston called HelpMap. They're part of this project, one of our partners. And they have this great system right now that you can currently use. It puts little pins on all of the outbreaks or all of the health-related things happening throughout the world in real time. So as part of their site, we were able to put in our own data. And so we put in all of the data from all of the surveillance that we do, and also after it's approved by the governments in each of these countries, we can then put the testing data. And you can see here, you probably can't get a sense just from this, but with each of these countries, each of those dots, you can then drill down into them. And you can see specific data where the sample was collected down to a village level, and then see the results from those tests as they're approved for release. And once we detect these viruses, although the science is in its infancy right now, we analyze them to the greatest degree that we can to try to get a sense for the risk of spillover. And here I've just listed some of the both virus traits and virus independence traits that have some sort of role in spillover and can help you to predict whether or not this virus has a capacity for spilling over or is a higher risk to humans. And some of these traits can be, if a virus, for instance, if we find it in a variety of species, then it shows that that virus is better able to cross over between species and therefore may be a bigger risk to crossing over into humans. There are also certain genes that we can look at to see if the virus is more pathogenic or more likely to infect humans. But in this next phase of the project, we're moving to expand this side of the science, which needs to grow quite a bit so that we can get even more predictive. I failed to mention earlier, but this project's been ongoing for 5 years now, and we were just renewed for another 5 years. So it'll go on for, 10 years, but hopefully more than that. But also our approach and our data in the laboratory is constantly feeding back into our field surveillance activity. So we're adapting and evolving based on the data that we're getting in the laboratory. So for instance, if we find an interesting new virus in a bat, we can then look back at the sites where we're doing surveillance, and we can look at all of the other animals in that location and see if it's just restricted to bats or if it's in a variety of species. We could also then find all of those bat species across the world and see if we can get a sense for the range of this virus. All of these things allow us to continuously refine our strategies in the field. And to date, we've been quite successful. The work's paid off. We've identified hundreds of new viruses. We've collected a large number of samples, and we've documented jumps of viruses from from wildlife into humans. We actually documented one human into wildlife as well, but that's a different story. This just shows some cool pictures of viruses and outlines basically some of our discoveries, and I'll go over a few of them in a little bit more detail. But you can see that we identified various new and divergent viruses, including paramyxoviruses and coronaviruses, some of those viruses that you see in the news that previously have been associated with human outbreaks. In addition to that, we've discovered a novel retrovirus. So retroviruses are like HIV, so it's important to pay a lot of attention to this group of viruses, given that one of the last times that they crossed over into humans, it didn't go so well. And we've also detected a novel clade of Ebola, and in some of the side work, novel primate lineages of malaria parasites, which have given us an idea about the origins of some of the malaria parasites that infect humans. So overall, we've detected in the last 5 years, or really the last 2.5 years since the first half of the project, we were setting everything up, we've detected a total of 984 distinct viruses. In total, 815 of those were new viruses. So essentially, this project's resulted in more viruses detected than the total number of viruses previously recognized in mammals by the International Committee for Taxonomy of Viruses, the ICTV, which is the international organization that names and distinguishes viruses as new viruses and adds them to the list. We cheated a little bit there because the last time they put the report out was in 2009, but we still found more than they had in the previous list. We'll see what the next list comes up with. Just to highlight a few of the discoveries. So we discovered the primate reservoir of a human retrovirus. So right now there are 7 human retroviruses, and all of them but one, we knew the source in wildlife. And so when we started to test in Cameroon, we found that 6 gorillas, lowland gorillas, came up positive for a virus that was very close to the human retrovirus, human T-cell lymphotrophic virus type 4, which I'll just call HTLV4 from here on out since the name is hard to pronounce. But you can see if you look, maybe here's a good time for the laser. If you look very carefully here, you can see a little red circle, a little red star. And this is the human case in Cameroon of this retrovirus. And you can see around that human case, you have a bunch of red circles. And these are the gorillas that tested positive for this closely related virus. So it starts to set up a very nice story here. Additionally, when we look back into the data that we had on this individual, he had indicated that he had hunted gorillas. And so this shows that there's still a very real threat of retroviruses moving over into humans, and it may be happening a lot more frequently than we thought they were. It's possible that, it's very likely that HIV started with these very early stage 2 or stage 3 transmission events before it learned to transmit between humans. Additionally, some of our other partners in the PREDICT program detected coronaviruses that were related to the current human MERS outbreak. And I'm not sure if all of you are familiar with MERS, but it's a lot like SARS. It was in the news for a while, and then the Ebola outbreak occurred, and so they haven't really reported much about it. But it started in Saudi Arabia, and it spread to a large number of countries since, and it's still ongoing now. And the source of this outbreak is still somewhat unknown. But at the time that the first few cases presented themselves and we had some sequence data, we looked into all of the sequences that we had collected for coronaviruses as part of this PREDICT program. And we found, as you can see, maybe I'll go to this side. No, trees in the way. We found that one of our coronaviruses, so here's the human case, one of our coronaviruses from Mexico, from bats, was very closely related to the human case. So this allowed us to suggest a role for bats in the transmission of this, in the cycling of this virus in nature, which allowed for more informed epidemiological investigations and also gave some guidance as to possible control methods during this outbreak. There's been some talk now that camels may be involved, but it still seems that it's possible that bats are involved as maybe the wildlife reservoir that then transmits down through domestic animals or something along that line. Next, some more work that we did in Cameroon demonstrated the first spillover of Hennaviruses from humans from bats into humans in Africa. So I mentioned Hennaviruses earlier, and this was the model virus that was used for the Contagion movie. they have very high mortality in humans, broad species tropisms. They really have a knack for crossing over. But previously, it was thought that they were only a risk in Asia and Australia. Although in the last few years, a few viruses were detected in bats in Africa, suggesting that they might have a broader range than was previously thought. So when we started testing humans and bats, we found a small percentage of humans were seropositive for this virus, which means that at one point they were exposed to it, even though they may not currently be infected. And we found about half of the bats that we tested were seropositive for it. And interestingly, we had a lot of data on the individuals from which we'd collected these samples. And we found that all of the individuals who were positive had reported butchering bats. Other individuals had also reported hunting bats as well, but in our analysis, hunting bats didn't come out to be a risk factor, but butchering bats did. It made you 29 times more likely to be positive. And one of the reasons for this, we think, is that in Cameroon, bats are hunted with rifles. And so when you shoot a bat with a rifle, there's much less blood release. It's just, hopefully, if you're a good shot, just one hole. But butchering it, then you're exposing yourself to all the bodily fluids. Additionally, we went and looked back to where all of the humans were positive. And we overlaid the map of the positive human cases with some recent data that came out from NASA that showed landscape change across time over the past, I believe, 2 decades. And we were able to correlate the individuals who were positive with areas that were recently deforested. So individuals who live in areas in Cameroon that was recently deforested had a 10 times higher likelihood of being positive for this. So overall, this adds to the evidence that land use change, living in this buffering zone, and also butchering animals leads to a higher risk of crossover of viruses. And now the discovery of Hennavirus crossing from bats into humans in Africa suggests that also in this continent we need to be paying attention to Hennaviruses as a possible source of infections in humans. It's quite likely that humans have already been infected with this and shown symptoms, and they were mistaken for malaria or some other disease, as a lot of acute febrile illnesses in Africa go unreported or unidentified. And just to talk about one of the human results that we have, I'm going to talk about a novel virus that we found that was associated with a few cases of acute hemorrhagic fever. So usually I'd have to give a lot of background on viral hemorrhagic fevers at this point, but I think just from a week or two watching CNN, you're all experts. But just as a little backgrounds, they're really bad. It leads to hemorrhagic type symptoms, so bleeding generally starts in the mouth and the gums and can lead to many other orifices later on. But they have a very short incubation period, abrupt onset, and oftentimes rapidly progress to death. Viral hemorrhagic fever agents are among the most pathogenic viruses that are known to man. But what's important for this next slide is, or the next few slides, is to let you know that to date, prior to this study, there were only 4 virus families that were associated with this type of syndrome. And those are arenaviruses, bunyaviruses, filoviruses, which include Ebola, and flaviviruses. So in the Democratic Republic of Congo, in a small village called Mangala, there were 3 cases of hemorrhagic fever. The first 2 cases were pretty close to each other, and they were both teenagers. They went to the same school and lived near each other. They arrived at the hospital and died within about a day of arrival. No samples were collected from these individuals. About a week later, the nurse that was caring for them, a male nurse, developed similar symptoms. So at this point, the staff were alerted to something potentially quite dangerous, and so he was quickly transported to a regional hospital in a city called Boma. And there they had the capacity to collect blood samples. So they collected a few samples from him, and they sent these out for testing in a regional lab in Gabon that we work with. And they tested them for all known hemorrhagic fever viruses. So Ebola, Lassa virus, Marburg, many of those that you've heard of, all of them came back negative. So we sent the sample to one of our collaborators in San Francisco, after appropriately inactivating the virus, should there be a virus in it. And this lab is the lab of Dr. Charles Chu at the University of California, San Francisco. And he did deep sequencing on this sample, which I mentioned earlier. A sort of shotgun sequencing approach, unbiased, detects all nucleic acid in the sample. And on our first pass of the data, we found a single fragment of nucleic acid that looked like a novel rhabdovirus. And after a little bit of time spent with the bioinformatician, we were able to de novo reassemble the entire genome of a novel rhabdovirus. So when we compared this to known viruses, it had very low similarity. In fact, it only shared about 41% of its genome with any known virus. And it was most closely related to arthropod-borne rhabdoviruses in Australia. So arthropods are ticks, fleas, mosquitoes, or biting flies. And in this case, these viruses were found in biting flies in Australia. So it's quite possible that this virus could be transmitted by biting flies in DRC as well. But further work will have to be done in order to demonstrate that. We named this virus Congo virus, even though at first we were thinking of Mangala, but these days you don't name it after a village because tourism gets driven down in that area, although Mangala isn't a big site in DRC for tourists anyways. So this virus and the rhabdovirus family now appear to be joining the viral hemorrhagic fever club. There is a precedent for rhabdoviruses to cause hemorrhagic syndromes, but only in fish. In human infections, all rhabdovirus appear to cause flu-like or encephalitis-like symptoms, like rabies. Rabies is a rhabdovirus. So this study shows that by paying attention to some of these unknown illnesses in these locations, we can discover new human pathogens, and hopefully that can help us prevent some of them. So in addition to detection, we also have been working on responding to outbreaks. Since we built up this capacity in all these areas, about 20 countries throughout the globe, we're well poised to also help out should an outbreak occur. I just have some pictures here of our lab in Yaoundé. This used to be an empty room, and if I were to blindfold you and fly you out to Cameroon and take your blindfold off in this lab for at least a minute or two, you might think that you were in a lab here in the Bay Area, at least until it rained or something like that, because they still drip a little bit. But you can see here it's a very sophisticated lab. We actually have whole separate areas for wildlife samples and human samples, so that you never accidentally call a wildlife pathogen a human sample or a human pathogen, and vice versa. And as part of our work, we've been building intellectual capacity as well. So we do all sorts of trainings, as I've indicated here. And also, we've been building capacity to respond to outbreaks. So we've standardized protocols for outbreak response. We developed a deployable laboratory, and I have some pictures of the deployable laboratory here. You can see on the bottom a picture of one of our deployable laboratories that was set up in eastern DRC during what turned out to be an Ebola outbreak. And this kit has everything necessary in order to both detect and then identify pathogens during an outbreak. Basically, a few big crates that you can throw into the back of a truck or into a plane. Just to go over a little bit of our success in this, recently there was an Ebola outbreak in DRC. It didn't get a lot of news because there's a much bigger Ebola outbreak in West Africa. But at the time, all of the members of the government who would usually be doing this testing were up in West Africa. And so they came to our team and asked if we could identify what was going on in eastern DRC. It was in a city called Bouende. And so my team members got all suited up, as you can see in the picture there, and they were able to identify the outbreak as being Ebola virus. And upon investigation of the sequence, the genetic sequence of the virus, we were able to confirm that this was a distinct outbreak unrelated to the outbreak in West Africa, which was important information to have so that we didn't think that the outbreak was spreading even further than it had. That outbreak, we got lucky, and it's now concluded. It was in a much more remote location, only accessible by motorbike or walking through the forest for a couple of days, so it's now over. We've also been in West Africa since 2009 working in Sierra Leone, and so we were well poised to help out once the virus, the Ebola virus, entered Sierra Leone. We've been helping out with diagnostic testing there. my lab manager on the ground there, I think might have tested more human Ebola samples than anyone else on the earth. I think her number is up to 1,500 samples. So she's currently up in France resting. The US CDC has taken over. And I'll just finish by saying, again, I want to emphasize that we're not using humans as sentinels. We want to prevent transmission events and the transmission of viruses from wildlife into humans. And so we take a lot of time educating the individuals that we work with to reduce their possible risk of infection. There are some really easy things that you can teach people to do, such as avoid blood contact, wash your hands, wrap your prey prior to transporting it so there's less chance of it dripping on you. Avoid higher risk species such as non-human primates, and also just don't butcher if you have cuts on your hands. This is one of the posters that we created along with some of our other partners in Cameroon. It highlights some of the dangers of bushmeat hunting and butchering and goes through some of the possible mechanisms that you can use to decrease your risk. So most of what I presented for you today was funded by Predict. We couldn't do it without them. And these are some of our other funders. A lot of funders through the Department of Defense, since they've started looking at viruses like TARIS, catch them there before they get here. And then all of our research collaborators that are far too numerous to name here. So with that, I'll take any questions that you have.